I'm going to go all dark too. Sorry.
Is that what happens when you record?
What?
When you record, it goes dark?
Yeah. I don't know why. I've never, it's been a while since I've had to record. There we go.
How many amino acids are there?
Oops. 20 different amino acids. Oh.
So are we going to learn about each one?
No.
Just the groups?
Just the groups. Yeah. Sorry.
So left-hand amino acids are more common?
Yes.
So there's 5 groups?
Yep. All right, can I move on to the next slide? I keep flipping
yeah.
back and forth. I'm just, sorry. All right, so there are 5 groups. So the 20 different amino acids can be placed into 5 groups based on their characteristics. The first one is that it could be a nonpolar amino acid, R group. So it just means that really you don't need to know this. All right, I'm not going to quiz you like, oh, you come across amino acid that has an OH group. What kind of, charge is it going to be, or what kind of, I'm not going to do that. So just know that they do have 5 different classes or 5 different groups. So they can be nonpolar, they can be polar uncharged. They can have just a charged amino acid.
Do we really need like the equation?
No. No. Like I said.
Like non-polar.
Yep. Yep.
Wait, what does the R stand for?
That's the R group.
Oh, so okay.
Yeah. Aromatic amino acids, which means they're in a ring. So if you don't know what aromatic means, that means ring. Sometimes we see it with alternating single bonds or double bonds. They tend to show that like this. So here's my ring, and then it alternates single and double bonds. So this means it's aromatic. And then the 5th one is kind of like, well, if it doesn't fit these 4, we're just going to call it a special function.
So this number 5 doesn't actually look a name. It's just
no,
a special function.
Yeah. So an example of that is the amino acid methionine. And for whatever reason, in eukaryotes, the first amino acid for every single protein that is ever made is always methionine. So, yeah. If you see a lot of prolines in your protein, then you see a lot of kinks. Cysteine tends to link chains together. So I'll talk about the levels of structure here in proteins. In fact, that's where we're going to go next. There are 4 levels of structure in a protein molecule. But just recognize that the shape of a protein affects its function. So each amino acid affects the shape of that protein and how it's going to behave or carry out a job. So a lot of disorders out there in organisms are because the proteins are not folded correctly, or there's a mistake in the recipe of that protein. All right. So a diagram just showing different classes, showing some 3D models and some Lewis structures of what they look like, just to give you an idea. So they group theirs into 4. I think they combined the polar uncharged with the polar, but you can see that just there's lots of different shapes for varieties for that R group here. All right. So amino acids are linked together by peptide bonds. So when you have 2 amino acids, they're held together by a peptide bond. It's a covalent bond. There's sharing of electrons. We go through that dehydration reaction. So that means water will be released in the process. Now I know when we think of bonds, maybe we tend to think of them as like rigid, like ball and stick models, but that's not the case. Bonds are actually very flexible. They rotate, they vibrate, they kind of move like back and forth. So there's actually a lot of movement. But in this case, with the carboxyl group, when it forms that peptide bond, it's not as free as all the other bonds. So it kind of behaves like a double bond, even though it's a single bond, and this just gives structural character to that amino acid. Now, when you have many amino acids linked together, now you have formed a polypeptide. So it's very similar to a polymer, okay? Because polymer just means a large molecule, but this one gives you an idea of which macromolecule it belongs to. Polypeptide, well, peptide bonds are only found in proteins. So peptide bonds are only found in proteins.
So, like, what we're going over right now is teaching us, like, why they're shaped the way they're shaped.
Yes. Because when we get to genetics, we will come back to proteins. So really this first unit is kind of like, what is science? Here's some chemistry. Let's talk about macromolecules. And then after that, it gets more biology-like. Chapter 4, I believe, goes into, I want to say, I was just working on it last week. But we'll get into like metabolism. Oh, cells. We'll start getting into different types of cells. Parts of a cell, prokaryote versus eukaryote. So next chapter you're gonna be like, oh, this is really familiar. Hopefully, right? Hopefully I did a good job sophomore year. Okay, so polypeptide. If you see that term, we are talking about a protein. It's just saying, hey, we have a bunch of peptide bonds holding amino acids together. Remember, there are 20 amino acids out there, so I'm pretty sure I have a multiple choice question that says like, how many amino acids are there?
20.
Yeah. Okay, so here's a diagram just showing the formation of two peptide bonds, of a peptide bond, going through that dehydration. So it's called dehydration because we're forming water. There's that O, there's an H. One of them should actually be an OH group. Okay, I don't know why they don't depict that, but it is an OH on one of these, and I don't know which one. But water will be released, and now we have formed a peptide linkage, and this is a very rigid bond, almost like a double bond, but it is single. It behaves like a double, but it's a single bond. Oops, sorry. So why is this important? So I told you that shape of a protein determines its job or its function. So I'm going to tell you a little bit about my research, which I might have mentioned back in Biology 10. So I know I say this a lot. But I was looking at a molecule called PP5 in circadian rhythms. So if you don't know what a circadian rhythm is, it's your 24-hour biological clock. You know, every organism pretty much has a circadian rhythm, even plants. So like for humans, you know, your coldest body core temperature is like at 5 a.m. So if you ever find yourself lying in bed early in the morning, you're like, holy moly, I'm really cold. Like that's always where your lowest body temperature is at. It's around 5 a.m. in the morning usually. You know, your first bowel movements are like 7:30-ish, 7 o'clock. And then your like optimal body temperature prime is like 1 o'clock. So if you have like some type of athletic event or some type of, I don't know, something where you have to like exert energy, 1 o'clock is like the prime time to do it. Because that's like when you're most awake and your body is ready to go. And then it starts to wind down after that. So like that's the circadian rhythm basically in a nutshell. But it is controlled by a bunch of tiny little proteins and molecules inside the cell. So this is my cell, and this is supposed to represent my nucleus. And this black line is supposed to represent DNA. And this is supposed to be like some type of receptor. And I know these shapes really don't make any sense, especially the terms GLOC and BAML1. But I was really focusing on CRY and PP5. So PP5 is a molecule that interacts with this molecule, which interacts with this. So you can kind of see that they interact with each other. Now, CRY is usually the starting molecule for your circadian rhythms. It gets activated by light. Light in red waves, red wavelengths, light in blue wavelengths. Okay, so light. And when CRY gets activated, it inhibits PP5, and it kind of just like can halt the cycle or start the cycle depending on the situation that you're looking at. But I was looking at PP5 and CRY and how these two interact. And so what we were trying to do was take CRY and mutate it. We were trying to inhibit it or stop it from binding to PP5. So we were trying to basically mutate the gummy bear so it wouldn't bind to cry. So how did we do that? Well, we analyzed the amino structure of it. So I know this doesn't mean much, but red means negative and blue means positive because the molecule we're looking at was pretty polar. So what we were trying to do was take one amino acid and change it. So here I took aspartamine 136. That means on the 136th amino acid of this protein, we wanted to take aspartic and mutate it. And in this case, I mutated it, and I don't know how I mutated it. I think I took off an OH group or something. Notice that it's red on this picture, but then down here, that blue is gone. So I was going to see if the electrostatic potential, like the difference, caused it to not bind to M prime. If that was the case, I probably could have won a Nobel Peace Prize, but I failed epically. Okay, like the two molecules still interacted, so it wasn't a big enough mutation. So I was in a class of 18 other individuals, and each one of us mutated a different amino acid, and we all failed. Actually, we wouldn't have won the Nobel Peace Prize. Our college professor would have taken all the credit. We would have been the minions. So, but yeah, just an example of, hey, we're trying to change the function of this protein by actually causing mutations, and charges do matter. All right, so non-amino acids, okay, they tend to hang out with other non-amino acids. So if you remember in biology, we talked about how polar loves polar. Like they will dissolve, polar molecules dissolve in other polar molecules, like cough medicine better be polar so it can dissolve into your bloodstream and go where it needs to go to help make you feel better. Well, nonpolar molecules like other nonpolar molecules because they hate water. They hate polar molecules. So they hang out together. A lot of times when you see a bunch of nonpolar amino acids, it can form rings or shapes based on those charges, really. So charges, like I said, they affect shape because of that polarity. Okay, moving on to the 4 levels of proteins. So proteins can be organized into 4 levels, okay? Remember, with our 20 different amino acids, they can appear in any position. So if I have a 100 amino acid protein, that means there's only 100 amino acids in it. That means these amino acids can appear in any order and they can double up. So I can take 20 to the 100th power and get a very, very large number and basically come up with a lot of different proteins. So an example of a big protein, especially important for us, is the hemoglobin, which is found in red blood cells. Hemoglobin is actually made up of 2 alpha globulin peptide chains and 2 beta globulin peptide chains. And these alpha and the betas, they do mean something. And we'll talk about that. I think it's a secondary level. Alpha means helix, and beta means sheets in proteins. But if you alter the sequence of the hemoglobin amino acid, you can change the protein. And this is where sickle cell anemia comes into play. Sickle cell anemia is just a 1 amino acid difference from normal hemoglobin, and as a result, instead of a nice little donut-shaped red blood cell, you get one that looks like a moon, crescent moon. Oops, what was that? Weird. Well, that was dumb of me. Why did I do that? So the first level is called primary structure. My bad. So primary structure is just amino acid sequence. What's the sequence of the amino acid? So if you come across a multiple choice question that says the primary level of a protein is, and you come up and you see options like hydrogen bonds, alpha and beta sheets, globular proteins, amino acid sequence. You would say amino acid sequence. That's what primary level is. It's just amino acid sequence. What's the sequence of the amino acid? So here's a picture just showing the primary level sequence of the amino acids. Okay. All right, now the second level, here we go. I was like, this doesn't make any sense. The second level of a protein, or secondary structure, is called hydrogen bond. So there are some H bonds on these amino acids, and they can interact with other elements like oxygen or nitrogen to form hydrogen bonds. Now there are two forms of secondary structure, and I mentioned it in the hemoglobin, the alpha. So if you see the term alpha in a protein, it means it's a helix. And the other one is the beta. We call that a beta sheet. So the protein hemoglobin actually uses both forms of secondary structure, alpha helixes and beta sheets, for its final shape. And these two shapes are formed from hydrogen bonds. So secondary structure involves hydrogen bonds that form helixes or sheets. So hopefully this next diagram shows you how they form these alpha helixes and beta sheets. Okay, so here's my alpha helix, and these are supposed to represent the amino acids, like the parts. So like there's the carbon, there's the H group, there's the amino group, there's the R group, and you can kind of see these dashed lines. These dashed lines are holding that helix shape. Okay, so they're like interacting north and south to form that helix shape. Or maybe if you flip the helix onto its side, but you get the idea. These dashed or dotted lines represent hydrogen bonds. They can also form a sheet where it kind of looks like kinks. Okay, so we have a string of amino acids this way and then this way, and they are forming these hydrogen bonds, and it forms these little kinks. So these are hydrogen bonds. Sure. So can I get back to you on that, because you're going to see it in tertiary picture. Okay, the third level is tertiary. No. Okay, so sheets are sheets. They're sheets. They're flat, but they go up and down. Kind of like, I don't know. It's like taking paper. You know how you fold it in little squares. You get so that's what I meant by kinks. So maybe I shouldn't have said that. I'm sorry. Okay, so folds and kinks. So secondary is primarily hydrogen bonds, just looking at how hydrogen bonds affect the shape of the amino acid sequence. Tertiary, we're going to throw in a whole bunch of other interactions, like hydrophobic interactions, where nonpolar molecules interact with each other. Van der Waals, which we mentioned. Nope, did we talk about van der Waals at all, chapter 1? I don't think we did. Oh, wait, yes we did. Did we read a paper about a gecko?
No.
No, we didn't.
Oh, shoot, that's right. That's right, the website didn't work. So I had to change it. Okay. Dang. Well, van der Waals forces is how geckos can climb up walls and hang from the ceiling without falling. So, shoots. Okay. And I shouldn't have done that. Okay. Disulfide bridges. Two sulfurs interact, and this is actually how people perm their hair. They add sulfur, and the hair gets all curly. So that's why you're not supposed to get your hair wet after you get your perm for like, I don't know, is it a couple of weeks? Yeah, something like that. So, Sosman's here, which is awesome. Ionic bonds, okay? Charges, negative and positive. So we have hydrogen bonds. Yes, I know, hydrogen bonds are secondary level, but they can be included in tertiary level. We have electrostatic attractions between positive and negative charges. We have that hydrophobic exclusion where if it's nonpolar and it's hanging out with another nonpolar amino acid. And then we can have the covalent bonds, like I said, with the disulfide bonds and getting your hair permed. I'm just going to make sure it is mentioned up here. The polarity. Okay, maybe it's quaternary structure when they talk about it. All right, everyone's got the 4? So, continued, just know that the strength of the attraction, it just like depends on the interior. So like, here's my amino acid sequence, and if I have like a whole bunch of nonpolar molecules, then I'm going to have a very strong hydrophobic interaction, where if I just have like a few, then it's not going to be as strong. But yeah, that's all this slide is really saying. So strength of stability is how well the interior fits together. How well can these forces hold that kink together? Yep. But it's not a what? Well, no, I'm not going to say that. It just depends on how many interactions are occurring in the middle. So like, understand that all of these forces are holding that kink together. So here I have a whole bunch of hydrophobic, they're all clinging together, so it causes that protein to have that curved shape. But then I may have an ionic bond further away from that hydrophobic interaction, and it's holding that kink as well. And then same with hydrogen bonds. So Okay, the fourth and final level of structure in a protein is called the quaternary structure, and these are the subunit arrangements. So I think this picture does a great job, and then I'm going to answer your anti-parallel with this diagram right here, Amber. So our primary level or structure is just the plain old amino acid sequence, okay? But the hydrogens that are hanging off of the amino acids can interact, and they can form alpha helixes and beta sheets. And that just means that they can be parallel or anti-parallel when you take a look at the tertiary structure. So you notice how like this is running this way, and then it loops around and it comes back. So that would be a case of anti-parallel, where one's going this way and this one, the other one's going this way. But then if you take a look, like, let's see if I can find one here. So if like this was to like loop around and then like maybe come back over here, this is interacting this way and it's going this way, so that would be a parallel instance. So it's just talent directionality. Don't get, like if you were taking a biochemistry class, then you would be quizzed on that. But your book is very in-depth, and I'm just going to go through it because you may read it and you're just like, I don't know what that means, so I'm going to tell you what it means. Okay, so secondary level is caused by hydrogen bonds. We get alpha helixes or beta sheets, but then we can throw in all those other interactions, like van der Waals, nonpolar, hydrophobic, covalent bonds, all of the ionic bonds, all of that. So then you get this really, really complex structure. But then if you have a whole bunch of these that can interact with each other, then you get your final protein shape, and that's the quaternary structure. So they color-coded orange to show, hey, that's this part. And there are 3 other tertiary structures that are coming together to form this final protein. Does that make sense? So like our hemoglobin is actually made up of 4 different subunits, 2 alpha, and then 2 betas. They said that. So that would just mean like, maybe 2 of these are alpha and the other 2 are beta. Whew. So quaternary structure, just looking at how those subunits are arranged for your final protein shape. So another diagram showing amino acid to our secondary structure, to our tertiary structure, and then just notice that this is just a small piece of the final protein. So, okay, moving on to, we're still in proteins, motifs, okay? Motifs and domains. So when we were discovering proteins, and we still are discovering proteins, sometimes we have no idea what this protein does. And so what we do is we look for motifs or domains. When you sequence DNA, in the beginning it was really slow and extremely difficult. And we know that DNA is directly related to the amino acid sequence, and so that helps. But, when we first were learning about proteins, it was a very slow and tedious job. Okay. So how did we figure out what proteins do? Well, we looked for similarities between a protein that we did know its job or function and compared it to unknown. So we're kind of like detectives. Hey, this shape in this protein does this. I wonder if it does the same thing for this new protein that we have discovered. All right, so I'm going to go on to define motifs here. So motifs are reoccurring thematic elements in music and design, right? But we can use the same concept in proteins. We can say a reoccurring protein shape or structure in its job, okay? But that's what a motif is in like music and arts. So like, for example, of some motifs that you may be familiar with, shaken, not stirred. It's pretty much said in every James Bond movie. That's a motif. Movie directors tend to have common characteristics or motifs. Like any Michael Bay movie, you better expect some explosions, right? He always has explosions in his movies, always, okay? Christopher Nolan really likes to mess with your mind, like Inception. And it's always kind of like dark, like, what was that war movie he just had, Dunkirk? Okay, so dark, and then he's always got like really interesting music. Like, he also was responsible for the Batman trilogy, so he likes to mess with your mind a little bit. Inception. Christopher Nolan. So, yeah. Alfred Hitchcock. He always had like a idea of a triangle in his film, where it was always a female and two males, and it was almost like a love triangle, but it was always thrown in a mystery or some type of dark, twisted story.
Like birds, like the birds.
Yep.
Can you go back one slide, Christopher?
Sure. If I wanted to throw in Star Wars, what's the line that's always said in every single Star Wars?
Your father, no, I got a bad feeling about this.
Yes! I got a bad feeling about this. Okay. So, what I'm trying to get across is that proteins have motifs as well. So, some common motifs that are found in proteins. Beta, alpha, beta. Also known as the Rossmann fold. So, here I have a beta sheet. It turns around, goes into an alpha helix. It turns around, and it goes into a beta sheet again. So, that's a beta, alpha, beta. We also have the beta barrel, which is depicted here. Whole bunch of beta sheets that literally form a barrel. So beta barrels are usually found in pores of cells, channels that allow things to travel through, because it literally looks like a tunnel. And then the final motif that I'll mention, the helix turn helix. So helix turn helix. Helix turn helix. I think out of all three of these, I would say the beta barrel and the helix turn helix are probably the most important. Helix turn helix helps proteins bind to DNA to turn on and off genes. Such a big section. Okay. So motifs, we use them in analysis of proteins. Really, that's all you need to know. We just use motifs to figure out what proteins do. So I'm just going to move on. We just use motifs to help us figure out what unknown proteins do. Now, I want to talk about domains. How do motifs fit into domains? So domains are on a larger scale. They're functional units within a larger structure. So I think your book used the term substructure within a tertiary. So if you want to think of your amino acids as letters, okay, the primary structure of proteins is the amino acid sequence. Your motifs would be words or phrases. Your domain would be a paragraph. A lot of proteins are made up of many domains. They can be physically separated, and they can also help proteins fold into their proper shape, which is where I'm going to go off to now. How do proteins fold into their correct shape, and what happens when they don't? I probably should answer this. Yesterday I was calling for a lot of photographs. Brittany Erickson. Good, but I think I've kind of figured out our bathroom situation, so I'm really sorry to waste your time. So, all right. No, thank you. Have a good day. Okay, bye. I just realized that's on a YouTube video now. Awesome. Yes, we figured out the bathroom situation. Okay, so here's a motif, like a beta, alpha, beta loop, and how it fits into the domain. Right? So Domains can help proteins fold. So the process of folding a protein relies on another protein called the chaperone protein. So we've made this protein, we got an amino acid sequence, we have all these interactions. But how do they fold correctly? How do they fold in the right way? How do they interact with those cross bridges within that same amino acid sequence? So the thought was proteins folded spontaneously as we have hydrophobic interactions with water and nonpolar amino acids in the interior. They literally thought proteins just did it themselves. But when we started to look at all those interactions, proteins can fold in so many ways. It would literally be a trial and error process, and it would take way too long. So how do proteins really fold? The answer is chaperone proteins. Okay, so how did we discover chaperone proteins? Well, they noticed that there was this unusual mutation in these bacteria cells they were studying. And these bacteria cells prevented viruses from replicating within them. And they're like, wait a minute, these bacteria cells should die. This virus has been affected, should kill off this bacteria strain that we're looking at, but there's some bacteria cells that are surviving. What's going on here? And they found that the normal bacteria cells had chaperone proteins that were correcting the error of these viruses. So here's a diagram of a chaperone protein where we have the chaperone protein right here. It's kind of like an open chamber. The mutated protein comes in, and it gets kind of reconfigured and folded correctly, and then it pops right back out. So chaperone proteins are just found in cells. They grab proteins, they fold them the way that they should. Don't ask me how they do that. And then they just release them. So in summary of this slide, we thought proteins did this. We thought they folded spontaneously, but it turns out they get help from chaperone proteins. So let's look at chaperone proteins in depth. Okay, they help other proteins fold correctly. There are many proteins that act as molecular chaperones. So I know we say, oh, the chaperone protein does it, but there are like many different classes of chaperone proteins. So an example would be if I were to take a solution that has an enzyme in it, and enzymes are proteins, and heat shock it, which means I just increase the temperature. Well, proteins are going to become unfolded, and it may not work right. But if I add some chaperone proteins to the mix, it will assist in the refolding, and the protein will be functional again.
Can you go back one slide for like 2 seconds?
Okay. So chaperone proteins, they just take misfolded or misprocessed proteins and they fix them so that way they can work right. So this native protein here just means that it's functional again. And when it becomes unfolded or misfolded, that chaperone protein will latch onto it and correct it basically. So some diseases are caused by improper folding. For example, cystic fibrosis. What's going on here is that a vital protein helps move ions across the cell membrane. So in a normal CFTR channel, we get chloride ions that leave the cell. And what chlorine does is it breaks down mucus buildup. But if you have a mutant CFTR channel, it doesn't work right. Notice it's closed up, like the chloride ions can't travel through and break down the nucleus here because it's closed up. So as a result, mucus builds up in their respiratory pathways, and it's not a pretty disease. Okay. Alzheimer's. Protein clumping in brain cells produces amyloid plaques. We believe could be caused by some type of protein deficiency where it's not folded correctly. Like the chaperone proteins are not there to help fold it correctly. So it's just one hypothesis. I don't know if that's true. Alzheimer's has made some milestones within the last couple years from your book being published. So that may be different. So when proteins become misfolded, okay, we tend to call them denaturation. So here's my normal protein. Oh, well, okay, we'll just carry on tomorrow with it.
Can you go back quick, please?
Sure.